Hi, I'm Laura Nairn. I'm here with Brezlin and we are here discussing the recent outbreak of the Labrador disease.
Right now, we are currently at the location where it all took place. Laura and I were playing fetch with the dog outside, Ellie, and we had threw the stick into the woods. Ellie had went to go retrieve the stick, and this is when Laura and I had discovered a crazy group of mushrooms we've never seen before. So when me and Laura came to retrieve the stick, we had found a group of mushrooms that were so unique. And Ellie had ended up eating this pile of mushrooms. So Laura and I had called the scientist and we asked for information about them.
A couple days later, the scientist came out to grab a sample and he took it back to the lab. The reason for concern is because Ellie is carrying 12 puppies in her belly, and so we want to make sure that those puppies are happy and healthy. 2 days later, in the middle of the night, Ellie had the puppies. So we brought her out here to this very barn, and this is where all 12 puppies were born. There were 6 girls and 6 boys. Me and Brezlin knew that by the end of the night, 12 puppies was way too many to handle. So we started calling our friends, and our first 2 buyers were Caroline and Dylan.
So after calling Caroline and Dylan, they had came over to the house for a few hours and loved on the puppies. The puppies were so excited because they thought they were getting a new owner, and they were right. After licking them and loving on Caroline and Dylan, me and Laura noticed something very weird.
After noticing the weird behaviors of both Caroline and Dylan, we didn't know what could have caused it, so we called the scientist to see if he had any results from the lab. And this is a video of what they were acting like. So the scientist gave us both the common and the scientific name. He told us that the common name was Labrador. Lab comes from the type of dog that Ellie is, which is a Labrador Retriever. And then the horn comes from the stinkhorn mushroom, which is the type of mushroom that Ellie consumed. Along with the common name, the scientist also gave us the scientific name. This scientific name is cani fungi. Cani comes from the word dog in Latin, and fungi comes from the word mushroom in Latin, referring to the stinkhorn mushroom which Ellie consumed, and Ellie would be the dog.
The scientific name of the disease is Boo fungi foeter. It originates from the stinkhorn mushroom, and it's divided into 3 ways, but it's not in order. It goes horn, mushroom, and stink, but it's converted in Latin. Horn means bucina, mushroom means fungi, and stink means foeter. The beginning of the name Boo comes from the beginning of the word horn, which is bucina in Latin. Fungi comes from the mushroom, and foeter comes from the word stink. These are all placed together to create the scientific name Boo fungi foeter. On the other hand, the common name of the disease is cogmissive. Cog incorporates the word cognitive because of the effect on the human's cognitive abilities. The suffix missive comes from the word dismissive. This explains why our friend Caroline was acting the way she was and wouldn't stop when we told her to.
The classification of our pathogen is both part of an animal and fungi taxonomic group. So the animal is Ellie, the Labrador Retriever, and the fungi is known as the stinkhorn mushroom that Ellie consumed.
So me and Laura were both confused by how Caroline was acting, but we made the connection. The scientists had told us that because Ellie wandered in the woods and ate the stinkhorn mushrooms, it passed through her placenta and passed on to the puppies that she gave birth to. All 12 of her puppies were affected by it, and all of their secretions carried the pathogen, the Labrador. So anyone who came in contact with the puppy would get the Labrador disease. If we were to come in contact with one of Ellie's pup's secretions, the secretion would first absorb into the skin. Absorption is the fact or state of being engrossed in something. The molecule secretions are so small that they are able to make their way through the matrix between cells of the stratum corneum, the outer layer of the skin, the epidermis. From here, the secretion molecule passes through the second layer of the skin, the dermis, into the capillaries, then lastly the bloodstream.
Once the secretion has entered the bloodstream, the short anterior communicating artery in the bloodstream, which joins the 2 anterior cerebral arteries left or right, moves the secretion to both sides of the anterior cerebral artery. The anterior cerebral artery is a vessel that's responsible for supplying blood to the front part of the brain, the frontal lobe. In other words, the anterior cerebral artery plays a crucial part in how Labrador makes its way from the bloodstream to the frontal lobe.
The chemical structure of our substance mimics the chemical structure of a neurotransmitter in the body, so it's capable of activating neurons. Once the substance passes the blood-brain barrier, the substance interferes with how neurons function via neurotransmitters in the brain. The signal is passed through the neurons that make up the brain in the same process as explained before. In summary, the anterior cerebral arteries send deep penetrating branches to supply the most interior portions of the basal ganglia. In the brain, part of the central nervous system, the basal ganglia is a group of nuclei interconnected with the cerebral cortex, thalamus, and brainstem. This is associated with a variety of functions, such as motor control, cognition, emotions, and learning. Dorsal root ganglia are situated outside of the blood-brain barrier. The deep penetrating signal sent from the anterior cerebral artery is received from the basal ganglia dendrites and then makes its way down the axon and at the axon terminal. With the same process as above, this signal is pursued through neurotransmitters. These neurotransmitters then make their way to the blood-brain barrier. The blood-brain barrier restricts the passage of the peripheral immune factors like signaling molecules, but Labrador made its way through the central nervous system.
On the phone with the scientist, he asked us what some of the signs and symptoms were that Caroline and Dylan were experiencing. He then explained that signs are what is seen and symptoms are what is felt by the patient. So some of the signs that Caroline and Dylan were experiencing were whenever they see a body of water, they just run and jump into it, chasing after things like balls, sticks, or toys. We then explained the symptoms that Caroline and Dylan were experiencing. These symptoms were wagging their tails, barking, running around on four legs, and licking people.
So after getting information about the Labrador disease, me and Laura wanted to make sure that we didn't get it, so we had to ask the scientist what we could do to avoid it. He said to make sure not to come in contact with any of Ellie's secretions, so this could be sneezing, drooling, or hair.
Now that Labrador has made its way into the brain, it has begun to affect the two cells that make up the brain, which is glial cells and neurons. Since Labrador has disrupted the cells of the frontal lobe, it has caused change in personality, mood, and thoughts. This is why the symptoms that are affecting Caroline and Dylan are causing them to go from human characteristics to canine characteristics.
After getting all the information we received from the scientists, we then put two and two together. The reason Caroline was infected was through the pup secretions. So this means that if Caroline got the Labrador, the pathogen disease, from the pup secretion, this means that it's indirect contact, meaning that it's spread through skin to skin.
A virulence factor is classified as something that contributes to an organism's ability to cause disease. The virulence factors that persist in the Labrador disease are adhesion factors, extracellular enzymes, and toxemia.
So now to describe each virulence factor. The first one we have is adhesion. This is what allows the pathogen to bind to the cell and take over. Secondly, we have extracellular enzymes. This is the chemicals that allow pathogens to invade further and persist in the body. The third virulence factor is toxemia, which is blood toxins found in the bloodstream. And there are two types. We have endotoxins and exotoxins.
After talking to the scientist, we wanted to return our friends back to normal. So he told us we had to go to our local veterinary office and ask for the cure to the Labrador disease. He then told us that there was an antibiotic that was being made as we speak. The antibiotic that was made is called bufungi transhorn, and this is an antibiotic that cures the Labrador disease, but there are some side effects. Just like any other antibiotic, this antibiotic has side effects. Mostly everyone recovers fully and has their human abilities back, but some people struggle with being able to talk to humans and interacting calmly with others.
In the making of this antibiotic, scientists had to make sure to use safety precautions. They wanted an antibiotic that would successfully get to its destination while not causing harm to the human along the way. With that being said, this is what we call selective toxicity. This is what efficacy is. So once the antibiotic has been taken orally and gets into the body, it goes into immediate work. It targets 6 specific places on a pathogen in order to take over it. The first one is the cell wall, then we have protein synthesis, the cytoplasm, we have metabolic pathways, DNA and RNA, and then the pathogen itself. After receiving the information we did from the scientists about the Labrador pathogen, Laura and I quickly drove to our closest veterinarian to grab the antibiotic, bufungi transhorn, and give it to Dylan and Caroline to return back to normal.